Dear students, in today's lecture we are going to talk about a very important environmental health and public health problem. Even though this seems to be mostly a public health problem, it has a very strong environmental component because this particular issue, which is the issue of antimicrobial resistance, and often it is called as antibiotic resistance, proliferates, that is, spreads and increases through environmental routes. And in fact, now we know that many of our human activities and our engineered systems are contributing to increase in the levels of antimicrobial resistance. Now, let us look at what is antimicrobial resistance. When we talk of antimicrobial resistance, we are talking about the ability of microbes to resist our attempts to kill them by using antimicrobials. So, antimicrobials are the drugs like antibiotics, antihelminths that kill our microbes, and we want to kill them because the ones we are targeting hopefully are pathogenic. But over time we have been noticing that these pathogens are becoming resistant, increasingly resistant to the drugs that we use to kill them. And basically the other way of saying this is the drugs we use for treating our diseases. So, at the beginning of antibiotics, when antibiotics were discovered and then made on industrial scale, the pathogens were susceptible or vulnerable to the drugs that we were giving them, the antibiotics. However, the question now is, why are they becoming increasingly resistant now when earlier they were vulnerable to antibiotics? And the reason is that pathogens in general were not resistant, but they have acquired the resistance from other microbes. Now, when we are talking about antimicrobial resistance, dear students, I must tell you that antimicrobial resistance in itself is not a novel phenomena. In fact, few years ago, a very nice study reported in a top-notch journal declared that even in an ancient cave that has not been touched by man or our activities, they found a wide diversity of antibiotic resistance in the bacteria. Why would that be so? Well, we know that many of our antibiotics and many of our antimicrobials are actually produced by bacteria and microbes themselves to use either to kill the competitor. So, only that particular microbe can survive and the competitor, or the ones it likes survive and the competitors die out. Or they also use these chemicals, which we call antibiotics or antimicrobials, as communication agent. So, bacteria and microbes, especially many bacteria we know, have a wonderful system of quorum sensing. In quorum sensing, the bacteria can sense each other's presence using chemical signals. They can communicate about food source and about threat, and so basically they talk to each other using these chemical signals. For many bacteria, these chemical signals are antibiotics and what we call antibiotics because they are toxic for some other kinds of bacteria. So, in this case, the bacteria achieves twofold benefit by producing antibiotics. First, it is able to talk and sense its own kind of bacteria, and the other, it is able to fend off the competing bacteria. So, antimicrobial resistance has been present since the inception of microbial life. Now, with human activities, we have enriched the levels of antimicrobial resistance, and also, as we talked earlier, microbes have the capacity to share genes, to share their attributes, and as the microbes that are resistant have enriched, they have also shared greatly their resistant genes, and this has eventually resulted in an elevated levels of antimicrobial resistance in our environment and also in our clinics. And as such, increasingly we have incidences of infections every year which are resistant to antimicrobials. So, let us look into antimicrobial resistance and the environmental aspect of it. Okay. So, how does antimicrobial or antibiotic resistance happen? So, typically when we begin with lot of germs. So, there are lot of microbes that are present in our community, environmental community or clinical community, and only few of them, the ones here in purple, are the ones that are resistant to the antimicrobial, to the drug. Now what happens is when we add, now let us say this is what is happening in a person's gut. So in our, this is the sample of our gut, let us say, and in our gut very few microbes are actually resistant to drugs, but they are not necessarily pathogenic. So we cannot say that this is pathogenic microbe, it actually could be both pathogenic or benign. Regardless of that, it might have antimicrobial resistance in it. Now when let us say this person falls sick and they take some antibiotics. When we take antibiotics, most antibiotics are quite broad spectrum, which means that they have the ability to kill a wide spectrum of different kinds of microbes, bacteria, and they are not very specific to the target pathogen that we want to treat. And such as what they do is they kill the good bacteria as well as the pathogens, hopefully. And now the problem is that these good bacteria were actually competing with the pathogens and keeping they were the internal system that was keeping a check on the pathogen concentration in the gut. But now because antibiotics have killed all these grey susceptible bacteria, now the body is more vulnerable to infections. And also we have killed all, but we have except for the microbes that are resistant. So now when the body will start being populated by gut, because now gut has a lot of food, so whatever survivors are there, they will eat it rapidly and they will divide. Eventually we find that we have the antibiotic resistance bacteria have overtaken the gut. So the good bacteria and the ones that were susceptible to antibiotics are dead, and the gut is full of antibiotic resistant bacteria. Now this is where the problem begins. Let us say this is our pathogen that is causing the disease, and let us say there was a pathogen survivor. Now these enriched microbial communities that all of them have antibiotic resistance or antimicrobial resistance will be able to transfer their ability to the pathogen. So this is what we say a benign environmental microbe. And now it was able to transfer via horizontal gene transfer its ability to resist the drug to the pathogen. And this is where the problem begins, because as pathogen feasts on the body, it will increase in number, and now no matter how much drugs I give, the pathogen won't die out and it will rapidly populate the gut. So these are all pathogens that are populating the gut now. The person will fall sick and more sick regardless of the person's dosage of the drug. Now this is how antibiotic resistance begins. Now let us look at how it spreads. So to begin to understand how it spreads, let's start from here. These are our antibiotics. In many countries including India, we have a very strong animal poultry and poultry farms. We have a very strong dairy and meat industry. Now in dairy and meat industry, we often feed a prophylactic dosage of antibiotics to our cows, buffaloes, goats, chicken. And the reason we do is this twofold. First, to prevent any infection that will reduce the amount of our poultry products and thus cause economic damage. And we also give them dose of antibiotics because now we know that farm animals that are fed, including chicken, that are fed subtherapeutic levels of antimicrobials. So subtherapeutical means the level of antimicrobial that is lower than the dosage. So if the chicken were to fall sick and it were to be given 50 milligram of an antibiotic, instead of giving it 50 milligram, I will give a much lower value of antibiotic like 10 milligram. So I give 10 milligram of antibiotics to my poultry animals, pigs, buffaloes, cows and chicken, and when I do that, I notice that their propensity to fall sick reduces and also they grow faster. So there are two benefits of giving prophylactic dosage of microbials to chicken. So there are two benefits of giving prophylactic dose of antimicrobials to poultry. One is that less chances of full blown infection. Which will hamper my economic profit, and the second is they grow faster. So, owing to these two advantages, we want to give a subtherapeutic dose of antibiotics or antimicrobials to our animals. However, following the same old logic that we talked about earlier here, that in a community when we give them antibiotics or antimicrobials, the susceptible organisms will die, leaving behind the resistant ones, and then the resistant will colonize the gut. And even if some other susceptible ones enter the body and start colonizing it, the resistant one would be very happy to share their resistant genes. So, over time what we notice is that the animals who are getting the antibiotics develop resistant microbes in their gut. Now, the other thing is, here we are giving example of George. So, this young hero takes antibiotics and develops resistant bacteria in the gut. Now, why would this person take antibiotics? What is the benefit for him to take it? First, he might be sick, so he wants to take it for treating the infection. Ideally, this is the case. We know what the disease is and we know what antibiotic would work well and how much dosage is required, and we give just that amount of dosage to the human being. But what often happens, especially in countries like India, where it is very easy to buy drugs. In fact, many antibiotics are over the counter drugs. So, if you feel like you are down with diarrhea or some other disease that requires an antibiotic, you can go to a medical store and buy antibiotics. As such, more often than not, more than 80% of time, we noticed that the antibiotics were not really required. For example, personally I know people who have had antibiotics for headaches, where clearly there is no link between headache and the any bacterial infection. So, as such, we are abusing the availability of antibiotics in our medical stores, in our market. And when we do that, we needlessly enrich the population which is antibiotic resistant in our environment. The other reason why George would take or the human being would take antibiotics is overdosing, overuse. So, for example, the doctor had prescribed the person to take antibiotics for a week, but the person decides, no, I am going to take it for longer time so that I am sure I do not get the infection. Now, the more we overuse it, we will see how later it becomes another problem, but the more we take antibiotics and the longer duration we take, the more good we are at selecting for only resistant microbes. And then the next thing we can do is abusing, overusing and misusing. Now, misusing would include conditions when the person really requires an antibiotic, but we are giving him a wrong antibiotic. So, the pathogen that was supposed to be killed by some other antibiotic is now developing resistance to this new antibiotic, and the good bacteria are dying. So, the infection is getting worse. So, abuse, overuse, misuse and genuine treatment of infection are the four scenarios in which antibiotics will be eaten by the human being. Often with cheap availability of antibiotics and poor awareness in the public, abuse, overuse, misuse are much more common than treating the infection. As such, over time the human being develops resistant bacteria in his gut. Now, here is the thing, now this person lives in the community, and when they live in the community they excrete, or wherever we live we do excretion, and that excretion through different portals will eventually land up in wastewater treatment plant, or will land in our drains, in our septic tanks, on our roads, on our fields, depending on where the human being is pooping and how our solid waste management is. And as such, we start spreading resistant bacteria in the community. So, the family members and the friends of this particular human being, after the resistant bacteria have enriched in his stomach, will also start getting exposed to resistant microbe, which will, if they are also abusing drugs, then their stomach, the good susceptible bacteria will die out and only resistant bacteria will be enriched. Now, the other thing is maybe the human being now is really sick and then goes to a nursing home or a hospital to take care. Now, when this person is being treated in the hospital, his samples are being collected, his wounds are being treated. Now, in this particular case, it is also likely that this person will spread disease to other patients. So hospitals is are very tricky and very sensitive location, because in hospitals we use a very high dosage of disinfectants and very high dosage of antibiotics. Most of the patients who walk into the hospital are on some or other antimicrobial, and because of that the people who come there have this kind of condition, which is the good bacteria have died out and the resistant bacteria are increasing. And also many a times people have surgical wounds or other wounds, and or we are collecting body samples, and if it is not being done carefully, then the person is likely to spread it to other patients who already have compromised immune system and whose good bacteria have already died out because of use of antibiotics. Now this person who has been overusing, abusing or misusing antibiotics will spread the resistant microbes to the other patients in the hospital, will also spread the will also infect or put the microbes, deposit microbes on the surfaces of the infrastructure in the healthcare, right. So for example, let us say I have a wound in my hand and that wound has a Staphylococcus aureus infection in it, and it so happens that I have been overusing, abusing or misusing drugs, or I am just unlucky enough that the Staphylococcus aureus is resistant already in any way, for any reason, but the infection now is resistant to the drugs that I was taking to treat it. Now if I go to hospital and the cotton gauge is used for cleaning my wound and then for disinfecting and other purposes. Now all these cotton gauge etc., they have the resistant microbes on it, resistant pathogens on it. If it is thrown in trash can or somebody mishandles it, or I just go and wipe the surfaces, so now I have developed fomites in the hospital. The floor, the surface, in fact even the door handle, the scissors, all of them are potential surfaces where the antibiotic resistant microbes and pathogens may have been deposited. Now let us say it is the door handle. When I am opening the door, I brush off through my wound or through my skin where I have some antibiotic resistant Staphylococcus aureus sitting, and when I open the door using the door handle, that microbe is antibiotic resistant Staphylococcus aureus pathogen is deposited on the door handle. Next time a patient walks in, and then the patient will pick up the infection. So this is fomite. The surfaces, when we touch them we fall sick because the pathogen has been deposited on them. So either way more patients fall sick, and when more patients fall sick, when they go back to their community or when they interact with their family members who are visiting them in the hospital, they make them also be exposed to resistant microbes. Now let us come back to our animals. Now these animals have resistant microbes in their gut. Now many a times we use their manure, their poop. For example, definitely in India we use cow dung as manure. When we use their, and also chicken droppings. So when we use their fecal matter for manure and fertilizer, what we are doing is we are depositing on our farms, agriculture farms, we are depositing antimicrobial, antibiotic resistant bacteria and pathogens also perhaps. Now as a result, the food we eat, the soil that is here is now getting populated by antibiotic, antimicrobial resistant organisms. Now if there is a rain event or if there is just irrigation event, then the water also will carry antimicrobial resistant microbes, which now can be transported through environmental routes, and they will eventually expose to other people also. Also the people who eat this food, let us say raw, will also get direct exposure to the antimicrobial resistant microbes. So this is an example of how antimicrobial resistance spreads. It all starts with overuse, abuse and misuse of antibiotics, which either in case of human or in case of animals. Now let us look at this here. So on the left side here we have a rough diagram on how antimicrobial resistance in E. coli, Enterobacter and Klebsiella, all three of them are potential pathogens depending on what kind of E. coli etc. it is. We notice how the resistance has increased over time. So there is a consistent increase in the resistance in E. coli, consistent increase in the resistance in Klebsiella and an Enterobacter. Now if you look, this is from 1990 to 2012. Now this particular diagram is showing how the number of new drugs in the market have been consistently decreasing from 1983 to 2007, and the picture ahead is even more dismal in the last 10 years. So this is pretty sad, as we are antibiotic resistance is increasing, but new antibiotics we are not able to discover them. This is bad because let us say if microbes are resistant to an old antibiotic, if we discover a new one, we can use a new one. I will give an example. Earlier in India, ciprofloxacin was a very commonly used prescribed antibiotic. Over time, the doctors found out that many infections were not responding to ciprofloxacin treatment. The reason being perhaps, and now we know actually, the reason was that the microbes, the pathogens were resistant to ciprofloxacin. They had acquired this resistance. Then we started using ofloxacin, which the microbes, even those who were resistant to ciprofloxacin, could not resist, and then we could treat the infections successfully. Then what happened with time was the microbes also acquired resistance to ofloxacin, and now we are using catafloxacin. So, we are moving from one drug to another just to play the catch on game with the microbes who are increasingly getting resistant to the drugs we are using. So, for this we require that as antimicrobial resistance increases, our rate of discovery of antibiotics should also increase, but that is not what is happening. If you look at this top left panel, antibiotic resistance is increasing, antimicrobial antibiotic new drugs are decreasing. Alright, this is another timeline of how we have detected different kinds of antibiotics. From 1928, the discovery of penicillin, to 1987, liptopeptides, and after 1987 our the trend has declined. So, see, no new antibiotics from 1990 to 2010 and forward, no new significant new drug that can be used as a successful antibiotics on a large scale without serious impact on health. So, now we do have the last line of antibiotics like vancomycin, etc., but they are, now we also have resistance against vancomycin, by the way, but they have serious side effects for the patient. So, we do not want to use them early on. So, you notice here there was a peak of discovery in antibiotics from 1940s to 1980, early 1980, but since then it has been readier. In fact, one of the guidelines of World Health Organization currently is to push the pharmaceutical industry to for research and discovery of novel antibiotics. Now, the question is, why would they not want to discover or spend money in new antibiotics when antibiotic resistance infection is increasing and there is a demand for new antibiotics? The reason is that drug design, drug delivery and testing requires drug design and its testing and approval requires lot of funding and lot of many years of research that need to be put in. And it so happens that right now this is not very profitable for the pharmaceutical industry to invest money in researching new antibiotics. Now, if you look here, I was talking about earlier how the major cause for antibiotic resistance in an environment are abuse, overuse and misuse of antibiotics. So, this is a very nice picture and this is a global information. So, what we have here is the total prescription in per year in millions, and if you note that the green one is the unnecessary prescription and the blue one is what was actually necessary. So, if you look at the year infection, we have 23 million in prescriptions per year, of which 70% are really genuine, they need the drug, 30% are additional, we can get rid of them. If you look at common cold caused by virus, now look here my dear students, if it is a viral infection, what is the point of giving an antibiotic, except that the antibiotic would get rid of the good microbes which compete against other pathogen and make the person more susceptible to further infection. So, this is 100% antibiotic prescription for common cold is excessive. So, we have 18 million excessive prescription every year. For bronchitis again, only 20% actually require antibiotics or they require help but not antibiotics. So, again here we have a very serious case of abuse. Sore throat, sinusitis, same thing, half of the cases it is not required. Now, in a country like India where pathology labs are not very accessible to majority of the public, the figures look even more grim than this. Okay, now let us look here. Now, we know that the microbes, when we eat antibiotics, they enrich in antibiotic resistant microbes. Now, if on a global scale also we note that the antibiotics for which we use more, we also have higher percentage of resistant microbes against them. For example, we have a tendency to use tetracyclines a lot. So, because we use tetracycline much more than we use gentamicin, chloramphenicol, this is chloramphenicol, this is gentamicin and this is tetracycline. We use it a lot more. So, the percentage resistant in the fecal strain are also very high. So, we have up to 80 to 90% of fecal strains that are resistant to tetracycline. On the other hand, gentamicin is not used very commonly, and thus the percentage resistance is also low. So, basically what we are hearing here in this particular plot is that if a community consumes one particular antibiotics at much higher levels, then the probability of finding microbes that are resistant to that particular antibiotic in the microbiome of the person is very high. Now, if you look here, penicillin, tetracycline and quinoline, over the years from 2002 to 2007, 5 years, within 5 years the percent resistance in Neisseria gonorrhea, which is a sexually transmitted disease, has increased quite some much, especially for penicillin and quinolines. Okay. Now, this is how does antimicrobial resistance, let us say the human being or the animal has been overusing, misusing, abusing the antibiotics and has enriched the antimicrobial resistance in its body. How does it transfer from one person to another, from one community to another, and nowadays as we are noticing from one country to another? Well, let us start from here. Humans. Humans are the ones consuming the antibiotics, that they are the ones that are producing them, and they are the ones following. So, let us say we have humans who fall sick. These sick humans will go to hospital. In hospital they get antibiotics, they become rich in antibiotic resistant gut microflora. So, the microbiome in their gut is now resistant to antibiotics, and ideally when they excrete, the excreta goes to wastewater treatment plant, which is not necessarily the case in India. In India, if we draw the same diagram for India, we will have one arrow going to septic tanks, and then the open drain. We will have another arrow going to field, open defecation, right. Then they will have their own diagrams. Okay. So, but ideally the excreta goes to wastewater treatment plant. Wastewater treatment plant also receive the household wastewater, which includes kitchen grey water and includes excreta, the fecal matter from bathroom. Now, the wastewater treatment plant, once it has treated the water, it releases the water in wetland, and there are recent studies showing that antibiotic or antimicrobial resistant genes, or the genes that confer antimicrobial resistance to microbes, do not die necessarily in wastewater treatment plant. In fact, many a times they get enriched relative to the entire bacterial population. So, now they go to wetland, and now the wetland will share this resistant microbes with groundwater, which the human beings will drink and fall sick. Wetland will also share with surface water, which humans might use as a dry drinking water source and fall sick. Then the wastewater treatment plant, the other thing that is produced is sludge. Now, the sludge is either applied in fields or it is dried and then burnt off. If it is applied in field, again it will affect the groundwater, which will affect human health. The other thing is, instead of using humans consuming the antibiotics, we also have, beside human consuming antibiotics, we have livestock consuming antibiotics. Livestock drop their manure, their droppings in the field, and then that affects the groundwater quality, and that affects the human. Same thing here, the livestock will swim in the surface of surface water bodies, like we have buffaloes enjoying the mud in wet mud near wetlands. So, now they will also infect or populate the surface water bodies with resistant microbes. Similarly, the groundwater, the field will eventually contribute to the groundwater contamination. As a result, we notice that wastewater treatment plant is our primary gate where we can put an end to antimicrobial resistance spread from the human perspective. From the livestock perspective, the easy way would be, because we cannot control the grazing of animals, and we should not, and how and where they poop is very hard to control. What we can do is we can put an end to prophylactic utilization of antibiotics for fattening the poultry and also for making sure they grow faster. Now, here is a wonderful diagram showing the importance of wastewater treatment plant in antimicrobial resistance. The wastewater treatment plant is a very unique place. Why is it unique? First, it is the community pool of fecal matter. Okay. So, what we get here are very rich microbial communities, that are gut microbial communities, because the entire community's fecal matter is collected together. The other thing and also because this is the case, it is very nutrient rich. Feces are very nutrient rich, bacteria love them. So, these microbes are perhaps very happy enjoying the food. The other thing is, when people consume antimicrobials or antibiotics, a very small proportion is actually absorbed in the body. Lot of it is and the one that is absorbed in the body, is some good portion of it is actually excreted in the urine. So we have urine and poop which actually do not, sometimes antibiotics do not transform even when they are excreted out. Wastewater treatment plant also receive antibiotics from excreta of human beings. Many a times people have bad habits and they actually directly pop the pill inside the pot. So they receive direct antibiotics. Now we have a very rich microbial community from the community in the community fecal matter. All of them are clinically relevant and also relevant to human health because they were from human bodies. Now we also have lot of antibiotics, and we also have lot of other xenobiotics. Now xenobiotics is a way of saying objects that life has not seen before. Xeno is the foreign, something that is foreign to life on earth. So we have novel drugs that life is not used to. Then we also have heavy metals, pesticides and other things. Now these things are important to write down because they trigger expression of antibiotic resistance. My bacteria might have ability to have proteins that give it antibiotic resistance, but might never express it. Now put heavy metals in there and it turns the regulation on, and they start expressing resistance to both antibiotics and to heavy metals. So this is why wastewater treatment plant is considered as the most important point and the gatekeeper for spreading the antibiotic resistance, because wastewater treatment plant will receive community pool of fecal matter from a large population. They also receive the wastewater from hospitals. Now remember, hospitals already enriched in antibiotic resistance, most of the people, and they are not very rich in other microbes. So the other thing that happens in wastewater treatment plant is that under inactivated sludge, we increase the biomass a lot. So the biomass is very much, and then what we have is that the microbes, they are in very close proximity with each other and they can share their genes very quickly and successfully very fast. So the resistance travels from a resistant microbe to a susceptible microbe very fast within a wastewater treatment plant. The other thing is industrial facilities, antibiotic facilities, what they do is they give it lot of xenobiotics, which again trigger promotion and promote antibiotic resistance. Next, we also have slaughterhouses that send their wastewater to a typical wastewater treatment plant. Now the issue here is that the slaughter waste often contains guts of the animals that have been slaughtered, and as a result they have lot of animal microbiome. And now we have a very funny condition in wastewater treatment plant where we have pollutants that trigger resistance in antimicrobials against antimicrobials. We have fecal matter, so we have human relevant microbes, which could be clinical pathogens also. The other thing we have is heavy metals and other contaminants, so all including pollutant. Then we also have animal fecal matter and animal microbiome. Now in the slaughterhouse we get animals from the farm animals which are being fed antibiotic resistance to fatten them up, to make them grow faster so that you can slaughter them faster. And thus not only do they once they are slaughtered do they affect the quality of water in wastewater treatment plant and how much antibiotic resistance genes it has, but also they do droppings on the grass, and when they drop on the grass we have land farming, soil, fish farms, river, drinking water, sea. So this entire chain that comes here. So in land farming we can do there can be leaching of antibiotic resistance to the ground. There can be children might be playing here, and when they play here they might get a scrap or two, and now there are antibiotic resistant microbes sitting here which might infect the children. The other possibility is that as I mentioned, if it rains, then not only percolation down the earth but also the runoff. Okay. So but the land farming will affect the soil, which might also affect the river. And then the wastewater treatment plant often empties itself in the river. So now all of these actually affect the quality of water in the river. Okay, now I have been talking about transferring genes, sharing genes, and if this is a revision from a previous lecture where we talked about different ways in which microbes share genes horizontally. One is transformation, other is conjugation, and then we have transduction. Transformation is when a bacteria opens a pore in its cell membrane to pick up an extracellular DNA. So, what they do is they pick up an extracellular DNA and then they are happy and their pore closes again maybe. The other is conjugation. In conjugation there are two kinds of cell, donor cell and recipient cell. So, the donor cell will now what it does is it opens the pillars, comes into contact and then donates the extra chromosomal material here that wanted to be shared to this. So, now what we will have is this will come here, or maybe at times this just extends here and this makes a copy of it. Either case this will acquire the quality present in this extra chromosomal plasmid. Many a times we notice that antimicrobial resistance is actually present in the plasmid. In that case it is very important for us to stop this so that the susceptible bacteria now do not become resistant. Next is transduction, which is carried through bacteria or through bacteriophage or viruses. We have talked in depth about them. Okay. And this is another thing I would like you to know is cross resistance, co-resistance and co-regulation, co-expression. So, in cross resistance what we have is if microbes have an ability to kick some other pollutant out of their body and that helps them survive, and they use the same mechanism. So, mechanism that helps them resist some other contaminant is also helping them resist antibiotics. This is cross resistance. So, these efflux pump allow cell to kick out the metal and they use the same pump to kick out antibiotics together. They are happy, antibiotics do not hurt them, metals do not hurt them. This is an example of cross resistance. In co-resistance, let us say metal and antibiotics, their genes that are resistant, that give resistance to metals and give resistance to antibiotics are in the same plasmid or are next to each other, same operon. When this gets expressed, this also gets expressed. So, if a microbe community is exposed to heavy metals and this is triggered, then that community will automatically become resistant to antibiotic R also. This is co-resistance. Now, we have co-regulation and co-expression, in which when proteins are helping to protect it from one particular contaminant, they also trigger proteins that will help it it against another contaminant. So, in this case the efflux pump is not the same, but they both are regulated and expressed together. So, dear students, this is all for today. In next lecture we will continue on our talk on antimicrobial resistance and particularly focus on how it pertains to India. Thank you very much.